Sweet. Thank you for the introduction. So yeah, today I'm going to be talking about me and Anna's master projects, which was optimizing eDNA sampling for detecting multiple aquatic invasive species. So I'll jump right in with kind of a layout for the presentation. So I'll start with some background about why eDNA, how eDNA can be useful. And then I'll jump into activity one, which is kind of my role in the project. When should we use eDNA for different aquatic invasive species? I'll hand it over to Anna, and she'll be able to talk about activity two, which is optimizing the lab and field methods we should use to collect eDNA. And then we'll end with some conclusions and recommendations and have some time for questions. So I'm sure all of you know, Minnesota has a lot of water bodies. These water bodies are really diverse. They're all throughout the state, and they're very important for Minnesotans for economic reasons as well as the vast cultural reasons. Unfortunately, a lot of these water bodies are threatened by aquatic invasive species. So you can see on this map on the left here, this is each of these red dots indicates a water body where one or more aquatic invasive species is present. And there are 800 water bodies that are listed as infested throughout the state of Minnesota. Some of the kind of common invaders is, you guys are probably aware, zebra mussels up here, spiny water flea. You can see me holding a large common carp, and then Anna down at the bottom holding a rusty crayfish. And so these species have kind of very negative impacts for the ecology of the lake, as well as can cause some human health impacts and kind of economic impacts as well. So we have so many lakes throughout the state. You know, we're known as the land of 10,000 lakes. How can we know where all of these invasive species are and what lakes they exist in? The kind of traditional way to do this would be standardized AIS monitoring. But traditional AIS surveys are quite hard. They're time intensive and resource intensive. So you can think of the two aquatic invasive species pictured here, zebra mussels and common carp. They would require a pretty different methodology to be able to sample these species. So common carp, you might be out setting nets or seining or electrofishing, while zebra mussels, you might be doing some plankton tows or some diving to be able to look for those. And so once you have, the number of aquatic invasive species that we have in the state, I think that on the infested waters list, there's definitely over 10. It gets really complicated to sample for all of these species at the same time. So if you go to a specific lake, you want to be able to look for all these species at a single time and kind of efficiently. And that's really hard to do with standardized AIS monitoring. But eDNA can help, and that's kind of where we come in. So eDNA stands for environmental DNA. So it's just the DNA that organisms give off into the environment. And it's DNA that we can collect through water samples. eDNA has kind of a lot of perceived benefits over traditional sampling. One is that it can be relatively inexpensive. Two, and I think this is probably the most important for aquatic invasive species, is that it can detect multiple species from different taxa, even when they're at really low abundances. So we want to be able to find these species when they're just invading a lake, just getting to a new habitat, and be able to provide some management at that early stage of invasion. So I think this is probably the biggest benefit of eDNA in the aquatic invasive species context. And then also, and I'll touch on this a little more later, it's eDNA sampling is really easy to do, and it's conducive to wide-scale monitoring. So it allows for us to monitor a landscape of lakes like we have in Minnesota. And you can see on this bottom photo here, I'm holding an eDNA sample. So it's really just a sample that we have. It's just a bottle of water that we've grabbed from the surface of the water. But eDNA is still a relatively new technology. So it kind of has really blossomed in the last 2 decades. And because of that, there's a lot of questions that still persist about how we can use eDNA for different types of organisms. So what we are looking to do and what we have done throughout this project is look at how sampling at different times of the year and in different kind of lakes and lake conditions affect our ability to detect eDNA. And that's the objective that I'll be talking about. And then Anna is going to be touching more on what are the specific lab and field kind of recommendations that we have to be able to maximize our ability to collect eDNA. So overall, what we're looking to do is provide recommendations to people that are interested in using eDNA to get the most bang for their buck, be as efficient as possible when designing eDNA surveys with different aquatic invasive species and different lake types. So I'll start with what we did for objective 1. We chose these 4 target species, and I mentioned them earlier, but we got spiny water flea, common carp, zebra mussel, and rusty crayfish. And we chose these species based on their kind of known harm to water bodies, as well as the fact that we have pretty good estimates of where these species are throughout Minnesota. And so we chose 20 lakes where we thought at least one or more of these species existed. And you can see each of these red dots. Each of these red dots represents one of those lakes. So these lakes are all throughout the state of Minnesota. And then at each of these single red dots, each of these single lakes, we visited it 5 times throughout the open water season. So this is kind of right before or right after ice melts, and then right before ice comes back on in the fall. And then at each of these times that we sampled a lake, we visited it, or we took 10 samples per lake. And you can see here a little bit of the physical lake characteristics that we have for these lakes. So we chose these lakes based on the species that we knew were present, but also kind of to maximize differences within lakes. So we chose, really large lakes like Lake Mille Lacs, and then much smaller lakes like Lake Phalen in Ramsey County would be an example of that. And as I said, these lakes have different numbers of known aquatic invasive species. So you can see Mille Lacs here with 3 has known populations of zebra mussel, spiny water flea, and common carp. And then on this bottom panel here, you can see our kind of hypotheses about when these species might be most detectable based on their life history. So spiny water flea, we thought they would be most detectable around kind of late summer, mid to late summer, when they kind of reach peak abundances and start hatching, start getting a lot more active. Common carp spawn in the spring. So we would envision them to be much more detectable kind of in between or around these 2 sampling events, the first sampling event and the second sampling event. Zebra mussels release their veligers into the water column and are at a planktonic life stage in the midsummer. And so we figured when all of these zebra mussels are reproducing, we would have the greatest ability to detect them around that time. Rusty crayfish was a little more, I guess, less defined than the other species. They have kind of molt periods, 2 big molt periods throughout the summer. And then something that I didn't include here, but they also get really active and they become much more like trappable and collectible kind of in mid to late summer. So we might expect any time throughout this summer zone that we might detect them a lot more. So jumping into some kind of basic results here, we detected common carp, rusty crayfish in about the same amount of percentage of our water samples, right around 10. Interestingly, we had new detections of common carp in 5 lakes, and we missed them in 0 lakes. So any lakes where they were known to exist, we got common carp DNA from those lakes. Rusty crayfish, we got a lot of new detections in 9 lakes, and we missed it in 1 lake where it was thought to exist. And this might be because the Minnesota DNR doesn't keep on their infested waters list a index of rusty crayfish. So it might just be that these lakes kind of had rusty crayfish and for a while, and yeah, there's no way of formally collecting that data. Spiny water flea, we had much more kind of trouble detecting. We detected them in 2% of our water samples, and we had new detections in 2 lakes, and we missed them in 6 lakes where they were thought to exist. Zebra mussels, on the contrary, we had the easiest time detecting them. We detected them in 18% of our water samples. We had new detections in 1 lake, and we missed them in 3 lakes where they were thought to exist. And so this is a really kind of useful way to frame the background of how kind of detectable these species are based on their species, and to be able to say, oh yeah, we found this species in this lake. But it doesn't really get to the question that we have about sample timing and when we should be sampling for these species. So getting into some of the modeling that we did, we fit a model that allowed us to determine how different factors influence our ability to detect these 4 aquatic invasive species. And these factors were day of year, which was kind of our key question. We're looking at when can we best sample these species, as well as the site depth, so that's where we're sampling in the lake, how deep the lake is right there. The stratification status, so that's if the lake is really stratified or if it's in a period of mixing, and then the water clarity of the lake. And based on the numbers that we get for our detection probability, we can actually calculate how many samples we would be required to take to get a 95% or a 99% probability of detection if the species is present in the lake. So I'll show some of those results here now. And I also just want to mention quick that this project is really designed to be able to answer these questions about how we can sample best for these species. And our goal wasn't to look for new species necessarily, but obviously that happened. So jumping right in to some of the results here, this is a graph showing the seasonal impacts on detectability. So you can see common carp is this red line over here. Rusty crayfish is the green line. Spiny water flea is the lowest kind of line here in blue, and then zebra mussels are this purple line. And so if you remember back to when I was talking about how we expected species to have varying probabilities of detection based on their life history, this graph fits our hypotheses really well about this. So common carp are most detectable in the spring, and then they taper off throughout the year. Zebra mussels get really detectable kind of in that midsummer period, and then taper off on either end. Rusty crayfish in the green here they don't have a huge peak or anything like that, but they slowly get more detectable throughout the open water season. And then right around August, they kind of peak for detectability and diminish after that. And then spiny water flea, as I mentioned, is our hardest species to detect. So they have really low detection probabilities out in the spring and the fall. And then kind of like we hypothesized right around July and August, we get a little bit better detection probabilities. And so, like I said, you can look at how many samples to take to be able to detect these species in lakes. And we can do that. So on this right side here, we have the number of water samples required to have a 95% probability of detection over the day of year. So I think what jumps out to me the most from this graph is that if you're looking for these 4 species, no matter when you're sampling, you're going to want to sample, or no matter when you're sampling, you're going to want to sample at the time when spiny water flea is most detectable, which is kind of right in the midsummer here, mid to late summer. So our sample timing is really controlled by our hardest to detect species. And then all of the other species are kind of much easier to detect in comparison to spiny water flea. And I can show some of the other impacts here on detectability. So this is water clarity. So this graph is set up in the same way, where we have detection probability on the y-axis, and then on the x-axis we have the clarity of the lake in meters. And so you see that spiny or zebra mussels get much more detectable as the lake gets clearer. And the other species kind of have much lower increases in detection probability. And even though, we'll leave it at that. Yeah. Um, and here we can see the impact of stratification on detection probability. So once again, zebra mussels are much more detectable when the water column is mixed. And you can see that spiny water flea kind of exhibit a similar thing where they also get a lot more detectable as the water column starts mixing. And to me, this is really interesting because these two species are the only species that we monitored for that have planktonic life stages. So when the water is really mixing throughout the lake and just getting moved around all over the place, the species with the planktonic life stages have much higher detection probabilities versus something like common carp and rusty crayfish that are kind of going to be littoral benthic organisms pretty much stay the same whether it's mixed or not, the lake is mixed or not. And then sampling site depth, this is the kind of easiest to interpret. For me, you can see that as we start sampling deeper and deeper, the detection probability for all four of our species starts to decrease. So from this, you would say, sampling at shallower areas probably might be useful, but it doesn't have a huge impact overall. So now I'm going to transfer it to Anna, and I'll let her speak about objective 2.
Thank you, Chris. Yeah, hi everyone. My name is Anna Totsch, and I will talk about the methods portion of this talk from the field to the laboratory. Next, please. So our overall goal was to optimize eDNA methods for improving detection probability of multiple AAS in freshwater ecosystems. Our research objectives for this objective 2 includes 1, to determine which eDNA sampling and processing methods, or the combination of the methods, maximize our detection of multiple target AAS across the water body types, and 2, to evaluate which quantitative method is able to detect our target nucleic acids in low levels expected during early establishment. So we can go to the next slide, please. Thank you. For our research, we went to 5 different lakes in the northern region of Minnesota with known AAS establishment, which has been verified by the Minnesota Department of Natural Resources. These AAS include zebra mussel, common carp, rusty crayfish, and spiny water flea. These lakes were selected based on the fact that our multiple species of interest were contained in a couple of these lakes, and they all contain different abundances of these AAS. As you can see, the St. Louis River estuary contains all of our species of interest: zebra mussel, common carp, rusty crayfish, and spiny water flea, whereas Pike Lake only contains one of the species of our interest, which is zebra mussel. Next, please. So our study design includes taking a total of 75 water samples per lake, sampling at 5 nearshore points and 5 offshore points. Nearshore points are defined as less than 1.5 meters in depth or less than 50 meters from land, and our offshore points are defined as greater than 5 meters in depth or greater than 250 meters from land. Our baseline samples are a 250 milliliter water sample filtered through a 1 micrometer pore size filter. This sample group will have a total of 45 water samples, with 15 samples being collected at each location: nearshore surface, offshore surface, and offshore benthic. Our experimental groups were sampled as paired test groups, which includes taking a 250 milliliter water sample filtered through a 5 micrometer pore size filter, and taking a 1,000, next please, taking a 1,000 milliliter water sample filtered through a 5 micrometer pore size filter. 9 samples from each of these groups will be sampled at nearshore surface locations, and 6 samples of these groups will be sampled at offshore surface locations. Please. So for impact of our sampling location, we wanted to know if there was a difference in frequency of detection between benthic, nearshore, and offshore locations. We found that where we sampled, there was no significant difference in detection probability based on presence and absence of eDNA. We used a one-way ANOVA to compare the means of the 3 independent groups in order to determine whether there is a statistical evidence that the associated population means are significantly different, and we found that they are not. Results are graphically displayed with the frequency of detection on the y-axis for location of samples taken, and species is on the x-axis. Only positive eDNA detections have been displayed in the graph. However, when running ANOVA in RStudio, we also counted non-detections. Next, please. When filtering water samples, we are looking to capture biomass harvest, such as cellular material shed by organisms through skin and excrement into the aquatic environment. By doing so, eDNA capture onto the filter can be super tiny, which is why the filter pore size can matter when looking for aquatic invasive species. For this experimental group, we ran a paired t-test for filter pore size between 1 micrometer and 5 micrometers. And we found that based on presence and absence of eDNA detection, the smaller pore size did significantly better than the larger pore sized filter. This makes sense as eDNA breaks down in the environment, and we'll want to have a filter pore that captures the eDNA onto the filter, then have it filtering through. Results were also graphically displayed with frequency of detection on the y-axis for filter pore size of samples taken, and species is on the x-axis. Only positive eDNA detections have been displayed in this graph as well. But however, when running our t-test in RStudio, non-detections were accounted for. Next, please. So similar to filter pore size, we are looking at water volume. And with a larger water volume, it could mean a higher probability of eDNA detection because of more biomass harvest capture. With this experimental group of the effect of water volume has on detection probability of the presence and absence of eDNA, we found that 1,000 milliliter water sample compared to the 250 milliliter water sample group had a significant difference in detection probability, favoring the 1,000 milliliter water sample group. This makes sense as well, as more water volume, the higher the probability of eDNA capture from the water. Results are also graphically displayed with frequency of detection on the y-axis for water volume of samples taken, and species is on the x-axis. Only positive eDNA detections have been displayed in the graph. Just like in our other t-test with the filter pore size, we also ran the t-test in RStudio, and non-detections were also accounted for. Next, please. So with the collaboration of Dr. Alex Badges, part of MACERC, we also are creating a generalized linear mixed model to find the predicted detection probability of the 4 tested AIS species under a hypothetical sample processing condition. Variables in this model include sampling location, water volume, filter pore size, and species, with some water characteristics as well taken at our sample sites. So next, please. And that includes clarity, dissolved oxygen, temperature, and conductivity. This model is still under revision for final touches, but results from this model is in agreement with sampling location, water volume, and filter pore size. That where you sample from does not matter in regards to detection of eDNA. The larger the water volume, the higher the detectability of eDNA, and the smaller the filter pore size may lead to higher detectability as well for eDNA. Next, please. So quantification methods can also influence whether or not you get a positive detection of eDNA in low quantities. Digital PCR is known to be more sensitive than conventional qPCR with absolute quantification and can maybe make a difference in practice because eDNA of AIS is expected to be in very low concentrations during early establishment. We tested if AIS eDNA detection can be improved by digital PCR by randomly picking 25% of the samples from activity 2 and running them on both qPCR and digital PCR. Based on the paired t-test analysis of detection frequency, the mean difference between the two test groups was significantly different from each other, favoring, next please, favoring digital PCR to qPCR. However, when the paired t-test based on eDNA copy numbers between the two groups, next please, the mean difference was not significantly different from each other. This suggests that more AIS detections were made using digital PCR when the population of AIS is expected to be low, but there is no difference between the two quantification methods when the population is greater or well above the detection of quantification. Gonna go next slide. So in summary, next please, a smaller filter pore size may be better capturing more biomass harvest. Next, please. And then a larger volume may be better at leading to more biomass harvest when a larger volume of water is being filtered. which overall can lead to more detections of eDNA for presence and absence. Next, please. Also, you have the ability of sampling from any location. Next, please. While utilizing digital PCR can potentially help you detect the low concentrations of eDNA found during early establishment of AS. These results make sense as DNA particles break apart in the water, and it may be captured on a smaller filter pore size. While with a larger volume of water being used, there's a higher possibility of eDNA capture within the sample, leading to more positive detections of eDNA for a species of interest. Based on the results of the pair-T test and generalized intermixed model, and also the ANOVA test, they are all in agreement with each other, which is very good to see. And that will be all.
Nice. Yeah. So just moving on to some discussion here. So as I mentioned at the beginning, the traditional monitoring of aquatic invasive species is time intensive and really costly. And because of this, there's a lack of statistically valid kind of monitoring throughout the state of Minnesota and an early response network. And we're not the only state that suffers from this problem. Pretty much any state with a lot of water bodies also has a lack of a kind of early response network for multi-species, multi-aquatic invasive species. But by using environmental DNA and combining it with traditional monitoring, we can better monitor AIS populations and spread over the landscape of lakes that we have in Minnesota. And we want to highlight here that eDNA works really well for most species, but there are organisms that are just generally harder to monitor. This was found through our results as spiny water flea, and this might be because there's lower spiny water flea biomass in the lake. They're giving off less DNA throughout their life stages, something like that. And when and how much to sample is dependent on the assemblage of organisms that managers are looking for. But the nice thing that Anna kind of pointed out is that the methods to sample these organisms are not species specific. So you don't have to use different filter sizes for different species. All of the things that Anna mentioned apply to all of the species. So I want to give a shout out to the continuation project that we have, which is started in 2023. And this continuation project sent eDNA kits to volunteer scientists throughout the state to be able to sample their lakes for aquatic invasive species. Currently, we've received all of these samples back, and we're working on the lab processing and generating data. So stay tuned for those interesting and fun results. Our general conclusions are that eDNA is a really new technology, and it's really has come to prominence in the last decade or so, and it's going to become an important piece of an aquatic manager's toolbox. The best recommendations that we have are to sample when a species is biologically active. So that's going to be when they're giving off DNA. And although this may sound kind of vague, it worked really well for us to be able to say, like, oh, we know that carp spawn in the spring. We think it makes sense to be going out during the spring to sample for them. And then based on Anna's work, as she mentioned, taking samples with a greater volume of water and using smaller filter sizes. So I just want to say thank you to our funders, partners, and supporters. MASERC has been really helpful through me and Anna's graduate tenure, and as well as the Environment and Natural Resource Trust Fund for helping us with funding. You can see both me and Anna's emails up there, so feel free to reach out to us at any time with any questions, and I will open it up to questions.
Awesome. This is very interesting. I'm super excited to hear more about this project. And I'm picturing you guys filtering a liter of water to 1 micron and knowing how long that might take. So, super, super exciting stuff. We do have a couple questions coming in. And the first one is there any work with AIS plants? And, is there any way that this can be implemented, almost looking at it commercially?
Yeah. So, I I think the reason we stayed away from plants in this is because one of our collaborators, Eric Larson, um, thinks that the primers and probes are kind of less developed, and it's really hard when there's hybrids and stuff like that to be able to use eDNA. It gets a little more complicated, but there are definitely people that use eDNA on plants, and there are labs that this can be sent to commercially. If you reach out to, I'll go back to my email here. I'd be happy to send you more information. I feel kind of weird plugging companies like on work time. But yeah, lots of places have started picking this up commercially.
Next question, thinking about, so looking at this from false positives, mentioning that you said that you didn't have any false positives. Is this first generally true with eDNA? And then second part of that question is if there's any drawbacks or trade-offs to using eDNA.
Coming from a lab perspective, false positives, they are something we always need to be cautious of. There's a wonderful paper that I've read all about false positives and false negatives. Really, the big thing on doing all the field work to the laboratories, you need to be very clean and sterile throughout each step. Use field blanks, lab blanks, like bleach everything down. That is what we did in the lab. So I feel pretty confident of the results that we have with commercial labs. I don't know exactly how clean they can get. I'm guessing they can get pretty clean doing this type of work. But also a big thing to remember is if you are having researchers coming from the field and going into a laboratory doing all this molecular stuff, you do not want that to happen. So I would be out in the field and I would go home and pretty much wash everything, then come back to the lab like in a day and just bleach everything down. So I feel very confident about our results. And yeah, so that's like the big thing about, preventing the false positives, I would say.
Yeah, I would say there's also multiple ways of looking at false positives. So there's the lab contamination, Anna, that you touched on, but then there's also like, what if a boater just has zebra mussels on the hull of their boat and they just give off some DNA? And that's kind of when it gets really hard to pick up. So we have all of these ways to measure if there's contamination in the lab, but there might just be a bird that came in from a different lake and pooped out some zebra mussels. Which has actually been a way that I think they have shown that zebra mussels have spread before. So, that's kind of a harder thing to get a handle on. And that's kind of why I mentioned at the end, it's nice to be able to pair these. So if you get a detection with eDNA, it's nice to be able to pair that with traditional sampling. So once you think a species is there, we can use this as a tool to recommend to people to say, oh, this lake might have, spiny water flea in it. We should go and survey that lake more intensely. So I think that's kind of one of the drawbacks of eDNA as well, is that it doesn't have these physical species collections. If you get a carp from a lake, you can say, okay, there's carp in this lake. If you get carp eDNA from a lake, that doesn't necessarily mean that there's carp in that lake. The carp could be coming from, eDNA could be coming from upstream upstream, but they might not be living in the lake.
Yeah, kind of one last aspect is to think about the traditional or physical methods that we have used in the past, or we are still currently using. And I also did traditional methods looking for the invasive species that we had listed, and I would probably say that there are a few more drawbacks in that, as we are looking for early establishment of AIS, which is why we're looking at more molecular methods. Once they are established, that's kind of when all the physical, detections occur, when we are physically looking for these species. It is also a lot harder to look for them, and having taxonomists on hand can be a challenge as well. So that's just a thought to put out there.
So we have a lot of interest in, you'd mentioned on one of your slides that you had some new detections, whether that have been for zebra mussels, spiny water flea, common carp. And so with those, thinking about those new detections, how confident are you in those results? And have you confirmed the presence in any way? Are you looking to confirm them? Have you worked with the DNR to potentially report those findings?
Yeah, great question. And this is kind of the hard part of this is that we get these new detections and we have to, provide kind of recommendations. So we have reported all of these to the Minnesota DNR. Some of these have since become confirmed. So we had multiple rusty crayfish detections on Mille Lacs, and we have seen on iNaturalist recently that someone posted a photo of a rusty crayfish in Mille Lacs. McCarran's Lake in Ramsey County was found to have had rusty crayfish detections based on our eDNA work. And I went out this summer and just did some trapping of crayfish. And we caught one singular crayfish the entire time we were trapping. And that one singular crayfish was a rusty crayfish. So we are definitely interested in continuing to verify these detections. Some of the species like rusty crayfish are kind of not have the management kind of objectives that spiny water flea and zebra mussels do. So it's, yeah, and they're just kind of likely to be more moved around through bait use and whatnot. So those are kind of have become widespread, especially throughout common fishing areas like the metro area. Yeah, so we are really interested in confirming these new detections and also working with the DNR to continue to make sure that we're having a high level of monitoring at these lakes. So one of the lakes that we had a new spiny water flea detection, we work with partners on that lake a lot, and they are going out and doing kind of this way of collecting spiny water flea in the sediment to see if they can find them in there, because that hasn't been done on that lake before.
How confident are you in those new detection sites?
Like I said, it's a little bit hard to kind of branch between if it is actually established in the lake. Like if, and when we were talking to the DNR, they kind of mentioned the same thing. Like if you find a dead zebra mussel shell at the lake on the shore, like near a boat launch, that doesn't necessarily mean that the species is in the lake. But this can be really useful as an early monitoring tool to say, oh, we have detections, we should go and survey that lake more. And so the specific species lake combinations that we're really confident in are the ones that have, I would say, multiple detections over multiple samples. And that ends up being most of the new detections that we got. But there are, the occasional time where you get one rusty crayfish positive hit on one lake. And I would say generally we're pretty confident in our ability to say if there is detections in the lake. It's hard to transfer that to, is this species active and living in this lake?
Yeah, and that's kind of the key thing of eDNA. It can tell us certain boundaries. It can tell us presence and absence. And I think that's the main thing. So even if there is a dead rusty crayfish in a lake body that we find, I wouldn't say that's a fail. I would say that actually is that the DNA, that the work we did, it did its job. That's how confident I am.
Awesome. You guys had mentioned that you had water samples that you had sent kits to volunteers. I'm just wondering how others can get involved in this process.
That is something that would probably be good to email and talk about later. It's kind of interesting because my advisor has a grand master plan of looking to do a lot more eDNA sampling for aquatic invasive species. But, that requires funding and getting more kind of partners on board. Reaching out to my email that I have on the slide right now would be great. Yeah, we had this project that occurred during 2023 that is kind of in the final phases. I believe there will be hopefully more of those opportunities in the future, but there aren't currently any right now.
Another question talking about water clarity. So I believe you stated that water clarity showed an increased level of detectability, especially when it came to zebra mussels. Could this just be because zebra mussels often increase water clarity, or is water clarity a good indicator for other AIS detectability?
Yeah, this is A very good question. The models that we're using, they're called occupancy models, so they can separate out reason a species is living in a lake versus the reason you are able to detect the species in the lake. And so we included water clarity as a detectability covariate. As water clarity increases, it's more detectable. The literature, I would say, is kind of mixed on this. Water clarity, like if there's really clear water, light penetrates deeper, and light has UV, and that can degrade DNA. So some people think that as lakes get clearer, you're going to have less detectability. But then there's some other interesting work that shows that if lakes are kind of brown, so this is really kind of particularly common near the northern border of Minnesota. The browning is because of dissolved organic matter, and this dissolved organic matter has PCR inhibitors in it that actually cause you to decrease detectability. So there's kind of different mechanisms at work here, and we are interested in looking at this further, and we do believe that the increase in detectability is partially attributable to clarity.
All right, I just wanted to mention too that for the few questions that are asking for lake-specific information, if you wanted to reach out to either Chris or Anna individually for that information, feel free to do so.
Yeah, and I see Doug put a really good comment in the chat also that there is a lot of early detection work that is going on at a local level. I would say this is like really great and obviously very important. This is like the kind of whole goal of MACERC. I think where it gets complicated is once you start putting in the multi-species assemblage of aquatic invasive species that we have in the state. So a lot of this work might be, monitoring for plants or maybe doing some netting or something like that. But to really get a whole kind of community look at what aquatic invasive species are in the lake, it's really hard to do that without spending a tremendous amount of time on each individual lake. So those surveys obviously have provided great new detections that we can make management decisions based on, but there are still many lakes that are going to be missed, and many species in those lakes that are going to be missed, even if there is kind of one traditional survey that is going on at a time.
All right, thank you so much, Chris, Anna,